Hello, my name is Jean-Paul Russo, and this is the presentation of my honors thesis poster. The title of my project is The Development of a DNA Single-Stranded Binding Protein Pushing Assay and Preliminary Results. Just as a bit of a background and introduction, single-stranded binding proteins, or SSBs as I'll refer to them as, play a crucial protective role in DNA repair and replication by protecting the single-stranded DNA once it's been unwound by the helicase from degradation. In recent research, more complex properties have been discovered in other species, most notably bacteria, and most specifically the E. coli species of bacteria. So the research aim of my thesis was to explore these complex interactions that SSB has been shown to have with key DNA replication proteins. In this case, the two proteins that I have studied with SSB are DNA polymerase II and the gamma complex clamp loader. The way that we set up our materials and methods in order to test these research aims was to specifically target the interaction between SSB and DNA polymerase II and target the interactions between SSB and the gamma complex. We accomplished this through the utilization of Forster resonance energy transfer fluorimetry, FRET. This is accomplished by having an acceptor fluorophore and a donor fluorophore. Basically, what FRET allows you to do is measure the distance between the two fluorophores as a function of fluorescence intensity. So my oligonucleotides, which are the substrates used in each of these assays, consists of varying fluorescently labeled DNA template strands, which have been annealed together to produce duplex DNA. Now, these fluorescently labeled DNA template strands, the top strand would consist of the acceptor and the bottom strand would consist of a donor, and we would use FRET to see how the addition of SSB to the assay enables those strands to either come together and move the acceptor and donor closer together or move the acceptor and donor further apart. 3 different separate assays were developed to test interactions between SSB and DNA polymerase II, or on the other hand, SSB and the gamma complex. And this data was analyzed by converting the values of fluorescence obtained through FLET fluorimetry to relativize that to the highest value of signal from the donor fluorophore. Moving into results, as you can see in the top panel here in the middle section, the first assay focused primarily on the interaction between SSB and the gamma complex. So these substrates consisted of a duplex created from a YA1 Psi3 3' donor fluorophore, which is on the 3' recessed end of DNA as the primer strand, and a JH9T65 Psi5 5' acceptor fluorophore on the 5' overhanging end as the template strand. As you can see in the graphs in the top right of the middle panel here, DNA substrates were shown to have a low FRET signal when analyzed independently. So all that means is that when we added the DNA to the assay by itself, it shows a low overall FRET signal, meaning that the two molecules are fairly far apart. Once we added the SSB in, as you can see, each of the peaks here on all 5 of those graphs dropped significantly, showing that there's been a significant quench of the donor fluorophore upon addition of SSB. Now, this indicates that the donor and the fluorophore are coming closer together as the SSB molecule is bringing the 2 strands of DNA closer together. After that, what we did was added the gamma complex in varying concentrations, 0 nanomolar, 20 nanomolar, 40 nanomolar, 80 nanomolar, and 160 nanomolar. And this is how we obtained each of the 5 graphs on the right. Now, what we found with this was that there was no significant change in fluorescent signal with the addition of the gamma complex. And I'll get into that a little bit more in the conclusion section. Finally, the next assay that we did focused primarily on the interactions between SSB and DNA polymerase II in a similar but slightly different manner to the first assay. So this is going to consist of a duplex with, again, a YA1 fluorescein fluorophore this time instead of the Psi3. And instead of using the wild type SSB, in this case, what we used as the acceptor fluorophore was an AF647 labeled SSB molecule. Now again, initially this shows similar results. The DNA substrates show low FRET signal when analyzed independently and significant quench with addition of the Alexa fluorophore 647 labeled SSB. With addition of DNA polymerase II, again, we saw something similar to what we saw in the gamma complex, which is that there was no significant change in fluorescence signal with addition of DNA polymerase II. Now, both of these results contradicted our initial hypothesis. Our initial hypothesis was that the fluorescent signal would be rescued or would increase with addition of either the gamma complex or DNA polymerase II. So just in conclusion, what we've seen is that increasing FRET signal will increase upon the addition of SSB. Now this indicates whether the DNA is in a 35 or 65 nucleotide binding mode. A 35 nucleotide binding mode would be where you only have one SSB bound to the single-stranded DNA, and a 65 nucleotide binding mode would be where you have two molecules of DNA bound, or two molecules of SSB, excuse me, bound to each DNA molecule. We did have a possible concern of environmental effects due to independent interaction between the Cy3-fluorophore and the SSB molecule. This is known as a PIFE reaction, and this is something that we haven't been able to account for yet, which has led us to believe that we need to further characterize these interactions and dig a little bit deeper and see what we can uncover in terms of mitigating that PIFE interaction. But as I mentioned, the lack of significant FRET signal upon the addition of the two key replication proteins, DNA polymerase II and the gamma complex clamp loader that we were testing, does contradict our earlier hypothesis that we made at the beginning of this research thesis. Potentially, if you were able to look very, very closely at these graphs, which is very hard to do on this poster here, we can see that the FRET signal is actually slightly increasing on the scale of dilution. So there could be a very minimal change in FRET signal based on the addition of the gamma complex or DNA polymerase II, but this isn't something that we were willing to characterize as a significant impact or a significant change in fluorescent signal. Lastly, I would just like to give a thank you to Dr. Linda Bloom for overseeing and mentoring this project, and specifically Elijah Newcomb for his assistance with the assay development side of this project. Thank you, and go Gators.